From Microbe TV, this is Office Hours for Wednesday, September 4th, 2024. It's it for summer. I'm Vincent Racaniello, and welcome to your viral corner of the internet. Tonight we have a special mini lecture. I'm going to explain the immunology behind COVID vaccines and boosters and all that stuff. Stay tuned. Tell us where you're from. We'll give you a shout out from your corner of the world. Meanwhile, I would like to thank our moderators for joining us tonight here on September 4th. Tom from Oregon, thank you so much. Les from California. Steph SF from San Francisco. Andrew from New Zealand. And Frank is here. I saw him. Yes, Frank from New York. Thank you all for joining us. Thanks everyone else for joining us tonight. Queue up your questions. Tell us where you're from. Let's see. Well, tonight here, I'm in New Jersey, and let's see, it's cooling for sure. It's cooling. 19°C at the moment, about 40 miles outside of Manhattan. 19°C, and the humidity's gone down. Let's see what it is here. 56%. So cooling off here as summer departs. Now, summer's not officially over. I realize that, but in my view, summer is June, July, August, and you zip through them like crazy, and that's it. And then September is a whole different mental state, right? Not that anything changes. It's just different. Well, mostly I think schools get back in session. They actually get back in session in August, but schools and everything is cranked up again. But I'm always cranked up, so it doesn't matter. Just I notice it from afar. Let's see. So Pete is here. Pete is from London. I asked him, what part of London? Oh, I don't know where that went. Well, that's fine. Tom is up in Oregon. Hot with regional smoke pollution in Oregon coast range. I have to be in the field on ecological restoration projects. So I'll be here later. Okay, thanks for popping in. Les says, people who discourage measles vaccination need a slogan, make measles great again. Yeah, there's just no reason to discourage measles vaccination. And as you see, it's coast to coast throughout the U.S. We have many, many cases. We're going to have many more. So we're all unvaccinated kids, and they probably feel miserable. And it's their parents' fault because the kids can't make a decision on vaccination. And once we get up into thousands, then we'll start seeing kids with encephalitis, which is really bad, long-term consequences and even deaths. So, you think you know what you're doing, but you really shouldn't be allowed to be a parent if you're not going to vaccinate your kids. Lisa is from Columbus, 77F. Jan is from Tucson, pretty hot out there still. I didn't know you could thumbs up even before the start. Yeah, you can. You can hit the thumbs up right under the video button, video window, I think. And that helps other people find us, maybe. Dagfinn is in Oslo, 2 a.m., 17°C. Wow. That's pretty warm. 28 tomorrow. It's going to go from 17 to 28. That's quite a rise. Anyway, thanks for being up at 2 a.m. Sean is in Tennessee. That is a state in the southern part of the United States. Will is in Changsha. He didn't write it here, but I know he's from Changsha. 38°C. Wow. 84% humidity. It's pretty intense. Pretty intense. Wayne is in Urbana. 77F. Abdul Aziz is in Saudi Arabia. It's not very warm. 26°, I guess, because it's the middle of the night or something, right? Is it always hot year round? Is it like always 100 every day, or is there a winter where it's like 90? 90. EG is from Mississauga, Ontario. 21C and clear. Thank you. I'm glad you like the podcast. Hello, Questella from Ohio. Here's Pete. Pete is from the inner north of London. I was trying to think of the name of that big grassy thing up in the north of London. Something Heath, right? I used to go to the NIBSC, which used to be up there. Yeah, South Mims, Potter's Bar, and it's right near this big grassy area. But I can't remember what it was. It's a very cool. People know what I'm talking about. Potter's Bar, isn't that a great place? So the NIBSC is the FDA equivalent of the UK. It's in Potter's Bar, Harry Potter's Bar. Kang is in Chicago, 22C and sunny. You see the cooling trend? Abdul Rahman is from Brooklyn. I don't know. Is that Cyrillic? I don't know. What are you saying there, Abdul? Anyway, 23C in Brooklyn. Hello, Jessica, who's in Ohio. Thanks for saying hello. 71F at the University of Toledo. Betty is in Washington Heights, New York City. SRR is from Paso Robles, California. Brian is in Montreal, 21C and very pleasant. ASV will be in Montreal next summer. The American Society for Virology. Hope to be there. I like Montreal. Airy Ozone, 60 in Asheville, North Carolina. Pretty chilly. Yeah, that's it. Although we get some warmer days in September, but it's pretty much it. Kip, thank you. And Laura, thank you for your support of science communication. Kip and Laura from San Francisco, 70F, no humidity. Junebug is in Virginia. Nice background. Love the decor. It's a little busy, right? You can see more now because I got a light. Put it on the floor. You can just kind of see the glow of the light. Which hand do I do? This one here. There's a tube light on the floor which splashes light upwards because Kathy Spindler said I was too dark. So I rearranged the lighting and I have this main light, which is now pretty much in front of me. So it's illuminating me and some of it's splashing on the background. And then there's that little color there. So it's not dark. Yeah, thank you. I'm glad you like it. I've been here since 2020. Well, earlier than that, but 2020, I got serious with the podcast here during the pandemic because I had to stay home. So I got good equipment. And now I've renewed it to a certain extent. Yeah, thank you. Joseph is from Ontario, Canada. All right, we got a question from Kang. I'll answer later. Peter, Boulder, Colorado. Claire is from the UK. Vanity Nutrition, our other moderator, joins us. Thank you from Long Island. Good to see you. Condit said he was in London in June. It's too bad he didn't hook up. It would have been fun. That's right. I didn't know that until recently also. Michael is in Mexico City, 23C. Rima is in Iowa. Hello, Rima. How are you? GST Bio. Jonathan, Encinitas, California, 27C. Making phage preps. Cool. kind of prep? What kind of phage? What are you gonna do with them? Well, you don't have to tell us. You could tell us what kind. I could guess, but it would be a wild guess. Phil is from Pullman, Washington. Yeah, I knew a guy from high school who was Phil Mixter, but I don't think you're the same one. Maybe it's a common name. Yeah, I love to chat with you folks. Um, I've accumulated knowledge over the years. I want to share it, and I like talking about virology and science in general. And it's a good venue to do that, right? I like it very much. Daniel wants to know if you can get contaminated by Mpox handling packages at UPS. That is a really good question, Daniel, because in a couple of studies that have been done, patients objects in patients' room are contaminated. Like the TV remote and various buttons and so forth. It has infectious virus on it. So it is entirely possible that packages are contaminated. Yeah. I don't know what the likelihood is, right? We don't know. We don't have any package associated infection yet. But if I were working there, I'd wear gloves. I know it's a hassle. It's hot, it's sticky. But latex gloves, because you might get virus on your hands, and then you're going to inoculate yourself. And that's not good unless, you didn't touch your face. But, even if you have cuts on your hand, it could infect there. So, yeah, I hate to say it, but I think that's a really good possibility. Now, it may be that we're being overly cautious, but given that the objects in patients' rooms are full of virus, um, I think it's highly likely. Yeah. Silent Runner is in Wichita, Kansas, 35C. Happy birthday. September 4th. Big 70. Yeah. I remember hitting the big 70 2 years ago. 2023. That really sucked. Yeah, that's a tough one to get over because everything else is, yeah, I'm still young. Although some people say I'm still relatively young. I guess when I hit 80, I'll be old. Right? Now it's all in your head how you look at things, right? I feel young. I'll run after you. I'll chase you. It's silly, but do things like this. Okay. Charles is in Lyndhurst, Ohio. A couple people from Ohio. Virus leading state. John is from Minneapolis, 78F. Rita is in southwest Idaho, where it's very smoky. Is that from forest fires? Laura, 50 miles from Manhattan. It's cool here. The window's closed tonight. Yeah, 50 miles. So you have 360 degrees to go. Well, not really, because you can't be out on the ocean. So you're either north or west or south or points in between. But you're not east. That's easy to sort out. Phil wants to know what's the rate of hearing loss. Yeah, it's about 10%. That's correct. Pete says there's a video podcast I watch sometimes. His partner is profoundly deaf from having measles as a child. Abdur says we've had a large outbreak of atypical pneumonia lately, mostly mycoplasma. So how did you message Dr. Griffin? Did you send an email to Daniel at microbe.tv, in which case, tomorrow's the clinical update. I can make sure that your question gets read if it's a question. A lot of people send comments, and so we have so many inquiries that I prefer to answer the questions unless it's something really unusual. So try and sneak in a question. Luis is in Mexico working on an antiviral for flavivirus. Very cool. Very, very cool. Frank is in Santa Barbara. Artemis is in Montreal also, 22C. Yuxing is in Hangzhou, still quite hot, 29C. Jefferson Space is in Miami. Tracy is in Chapel Hill, North Carolina. Oh, whoa, whoa. Marco's in Phoenix, where it is 109 degrees. By the way, speaking of decor, so you can see that there's shelves here, and then it's like you can barely see a white phage. That's from Mark Martin. It actually lights up. So I'd like to, at some point, put maybe order the viruses so you can see them better. Maybe put them up here and then put a light in so you can see. That's something I'm thinking about. Yeah. John is from the capital of Freedom, Florida, which I guess is Miami. Freedom. Hello, Dr. Bro Shropshire. Stellar content, second to none. Huge fan. Keep it up. Thank you so much. I never forgot the words you said. Thank you for your support of science communication, by the way. You said once, there are two kinds of knowledge. There's the kind you keep in your head, and there's the kind you know where to look for. You don't have to have it all in your head. That's just so true. You said that a while ago, and I always remember it. So thank you for those words, and thank you for your support and for listening. Really appreciate it. Maureen is in Northeast Ohio. There you go, near Hampstead Heath. That's what I was thinking about. When I used to go to NIBSC years ago, I don't go anymore. It was close to the Hampstead Heath, which is a beautiful grassy park, right? Really nice. Hampstead Heath. So I used to know a guy at NIBSC, Phil Miner. I really liked Phil very much. He had a nice kind of accent. I don't know where he was from, but I liked his accent very much. And he was a smart guy, fun to talk to. And I often would complain about names, the pronunciation of British names. And he would say, oh, well, why isn't it Arkansas then, not Arkansas? I forgot what I was talking about. Yeah, it's right. It's Arkansas, but it should be Arkansas, right? Oh my gosh. Cool. Mike and Lori are in Seattle. We're at 78F and SRR. Thank you for your support of science communication. Using Super Chat to push the YouTube algorithm for TWIV up, and away. Thank you. Super Chat, likes. So we have 140 people now and 70 likes. So you could up that. And speaking of likes, we got 130,000 subscribers on YouTube this week. Very cool, right? Very proud of that. I mean, it's slow as molasses to get there. 128, 129, 130. I'm still waiting for 1 million. Gonna figure out something to do that'll get us to 1 million. Something. I don't know what it is. Brainstorming. Anyway, thank you for your support, SRR. Kathy is in Vashon Island. Where's that? I mean, I could look it up, but where is it? Cheryl is from Santa Rosa, 87F. Abdul was typing in Russian. Privyet. What did he say? What did he say in Russian? Do you know, Rima? Good evening. Lori is from Tomball, Texas. It's been a while. Glad to be back. Farmer Norm is in Sydney, Australia. Wow. Far away. That's probably as far as you can get from here, right? Patricia loves Paso Robles. Yeah, it's a nice name. Also have very good vineyards there. I used to like their reds very much. And Patricia's in Washington, D.C. Oh, yes. Dobry večer. Good evening. Yes. I thank you. Privyet. I didn't get it in Cyrillic. Dobry večer. Večer. Yeah. That I know. I have a list of the few Russian words that I do know, but they're not in Cyrillic, right? Yep. Hat is in the San Fernando Valley. Now, you like the purple light? So it's a tube light that can be tuned to anything. And I do like the purple. I don't know if I'm going to try some other colors, but it gives a nice atmosphere. I subscribe to what I call retail therapy. Now, I don't buy things frivolously, but I don't always buy what I need right away. But then sometimes you need retail therapy to improve your outlook. Yep, Hampstead Heath. That's it. Exactly right. John, thank you for your support of science communication. Twin 180 with Mark Martin. Mark O. Martin. He's not Irish. Marco Martin. It's funny though. I don't know if Mark is here. He would be amused. Tona is from Long Island. Welcome, Tona. Now, Dr. Shropshire, I know, is from New Orleans. So another representative down south. G is from between the Detroit burbs and Cleveland, Ohio. Okay, thank you. Yeah, Encinitas is nice too, right? Encinitas. Markle is from the east end of Long Island. 14C. Whoa. Whoa. Well, this will be recorded, of course. You can always watch it later with all the comments, except the ones that are moderated, which is the way it should be. David is in Dayton, Ohio. So Kip says, my daughter says New York City was recently in hell's front porch and now false fall. Next is second summer and then actual fall. That's exactly right. That's what happens. So you get horrible heat and then suddenly it's cool. You think it's fall, but it's not. Like what we're doing now, this cooling, it's not. We're going to have a second summer where it's going to be hot. And then maybe in October, we'll get the actual fall temperatures. Oh, usually that's what happens, but there could always be exceptions. Shakir is from India. Kashmir, welcome. Very welcome. Yep, the humidity in San Francisco is currently 74%. It would be quite a feat to be surrounded by water on three sides and have no humidity. Ha ha. Ha ha. Not the same. Okay, you're not the same as my high school classmate. I was in Wisconsin. Okay. Yeah, I do know a Phil Mixter from high school. And saw him on online not too long ago, but not recently. Is there a way to accelerate viral evolution in the lab? So there are two components of evolution, right? There's mutations and selection. And so it's easy to devise selection. So you for example, you want to select a virus resistant to heat. So you can make more mutants by mutating. You could introduce more mutations into the viral genome. You can do that by mutating a plasmid copy of the genome, for example, with mutagenic PCR. And then you start with many more mutations, then apply your selection. You can look for monoclonal antibody resistance, but you can start with a broader mutant population that way. Yeah. So GSD is making lactococcal phage and Lactococcus lactis. Okay, cool. Yeah, yeah. Yes, I have said it, as I just noted before. Do zavtra. No, that's see you tomorrow. What is it? Now I've lost what it was. I'm not going to find it again. I'm sorry. Because I've forgotten what it was. I guess I could search for Abdur. Okay. Here we go. Wait, bear with me. I'm going to end up. Dobre večer. It's not on my list. Dobre. So that's good evening. It's not on my list. It's interesting. These are my Russian words for my stream. Anyway, thank you very much. Yes. Veronika's not here. Maybe not. Hat is in the San Fernando Valley. Oh, I already said that. You see, I got myself. Okay. Wearing gloves, Artemis says, will not prevent contamination if I touch my face. Yeah, but somehow the gloves remind you that you shouldn't touch your face, right? That's the hope. Because yeah, I don't know what else to do. You know, I could say wear a face mask, but you could still get it on your hands, right? I don't know. Do they irradiate packages? But you can't do that with everything because there might be something sensitive inside. Yeah, good issue. Yeah, good issue. Maybe later you can explain the polio drops. I saw some get drops younger age than others. Well, you need to get immunized in your first year of life, and a lot of the babies are not getting immunized, right? So they're getting them, and then I think the goal was to get 640,000 kids, and that can range in age, right? Depending on their vaccination status. If they don't know, then they will get vaccinated. But they're getting OPV, unfortunately, which will first of all, it will paralyze some of them, unfortunately, which I think is unethical. And it will circulate and continue to cause outbreaks of polio, right? You know, people tout, oh, we have eradicated types 2 and 3. So you still have vaccine-related types 1, 2, and 3, which are poliovirus, and they're causing polio. So it's not really much of a victory, in my view. Carol, our favorite nephrologist from Bakersfield, where it's 105F. Whoa! Did you see the Nature article on Brazil opening a BSL-4? No, I didn't. Cool. What city? I guess in Rio, right? Most likely. I may go to Brazil in December. Fingers crossed. Fingers crossed. Thank you, Abdul, for your support of science communication. Sam is in Oracle, Arizona. Age is a moving target. It's always 10 years older than your current age. Yep. Should we worry about the current outbreak of Eastern equine encephalitis? This is not unusual in the Northeast U.S., right? Especially in Massachusetts. Should you worry? Well, the vectors, the mosquitoes that spread it are already there, so climate changes really shouldn't have an impact on that. But it can be encephalitic, right? And that's never a good thing. So, as always, you should you should avoid getting bitten by mosquitoes, which is possible, right? But people like to wear shorts, and that's not a good way to avoid it unless you're going to use mosquito repellent. I am very lucky. Well, I don't wear shorts anyway, but I'm lucky. Mosquitoes don't bite me. Actually, they don't. I don't know. There's something about me they don't like. Maybe I have too much information. Okay, let's see. Who else is here? Alejandro is in Temple. Temple. I don't know where that is. Is there good news on an Ebola vax? Yeah, well, yes, we did it on Twiv. Maybe it hasn't published yet. It's like fabulous vaccine. They did an assessment in the what was the latest outbreak in? Is it DRC or Uganda? And there's quite a sizable outbreak. And vaccination was used. So retrospectively, they assessed, and it was like 80-some% effective at preventing disease. So yes, and maybe it's just coming out Sunday. So we're now on a week delay on Twiv. So we did talk about it. Yeah. Oh, Dr. Shropshire is in Birmingham. Sorry, I thought you were born and raised in the Big Easy. Okay. Good, Tigers. Thank you. So you're living in Birmingham, and you grew up in New Orleans. Thank you. Thank you very much. Brian is in Sydney, Australia. Sean is creating a neurotropic liposome stuffed with ATRX CRISPR knock-in. Interesting. Interesting. Kansas, Arkansas. Is that a French thing? I actually don't know. Right. So it should be Arkansas. Phil Miner always used to say, Vincent, should be Arkansas. There was some British word that I was making fun of. And I don't remember, but you would recognize it. And he said, okay, well, then I'll say Arkansas. I don't know what the origin is. Yeah, it's pretty good, 130,000 subscribers, right? When I started the channel, well, I couldn't get to 20 for years. I don't even remember when I started it, 2008-ish. I don't remember. But, I started it as my personal channel, and now it's not really. So there's not much I can do about that. It's Microbe TV now. It's all our programs there. Because you don't want to create a new one and start over. So, but then, yeah, the COVID really knocked us into the running. It got us to 100,000. We got our platinum plaque. Oh, Mandy says 19-year-old's getting his first Mpox on 9/9. How long does he have to wait for his COVID update? Well, Daniel would say you could get them at the same time, most likely, because you can get a lot of other vaccines at the same time. But you could wait a couple of weeks, or you could ask Daniel. Daniel at microbe.tv. I'll ask him tomorrow. Yeah, I want more subscribers, mostly because, there's a lot of misinformation out there and we're not reaching those people. But we have to figure out creative ways. I need to bring new young blood into Microbe TV and have it with ideas of how to do that. Mm. Kathy is in Washington, and I know it is the state of Washington because that's what Washington means. Yeah. Um, I did apply for memberships months ago when you first suggested it, and I haven't followed up. I should. I will. Brian is from Sydney, Australia. Vachon is an island in Puget Sound just across from Seattle. Harry also wants to know if Kathy is the author of the Abascal Way. Will, I have a little knowledge in my head and a lot on OneNote on my phone with a special section on Twitter. Yeah, well, after many years of virology, I have a lot of virology in my head, but more offline. Yeah, for sure. Absolutely. Retail therapy. Laura knows what I'm talking about. Keeps the drug companies broke. I went window shopping yesterday after paying all the bills. Yep. Best time to go. Yeah, but you pay the bills, you don't have any money left. Have you considered the historical significance of TWIV from January to say May? Yes, it is an archive because for the first year, every week we had at least 2, sometimes 3 episodes on the pandemic. So it is a wonderful archive, but I don't know what to do beyond that. Any suggestions would be welcome. All right, so Pete has some more history. And Hampstead Heath was preserved from development by an 1871 Act of Parliament. Long story on attempts to develop in previous decades. History is on Wiki. Now that I've jogged my memory, I'll go check it out. Hampstead Heath. Yeah, it's so cool. I think it's interesting when you travel, you find out local stuff that people who live there know and not elsewhere. But so. All right, who else? Bonnie is from Milwaukee. Do you need to still be masking? Well, it's up to you. If you have a health situation that would make COVID very bad, then you should. Yeah, when you're amongst a lot of people, right? So any immunosuppressive state, heart disease, diabetes, and many others. Or now here's the other thing: if you're over 65, you're at higher risk for getting severe COVID. And so one option is to mask, one option is to be ready to take an antiviral or have frequent vaccinations. But as we'll see, the vaccinations are not going to prevent you from getting sick. So, I still see people masking. I think if you are at risk for severe disease, it's not a bad idea. A book, documentary, archive for history. Gosh, I think they're good ideas. Yeah. I don't have the bandwidth. Someone would have to do it. And we are a niche thing. So nobody's going to do it. They'll say, nah, there's no money in it. Can the Mpox survive on the surface of a package through a transatlantic flight or in ship containers? What's the half-life of the infectivity? Let's take a look. Mpox infectivity half-life. This is where Condit would be helpful here, right? Well, unfortunately, it can retain infectivity in the environment for days to weeks under certain conditions, can survive in scabs for months to years, can be detected on household surfaces for 15 days. So it is inactivated by common disinfectants and acidic conditions. So, but yes, so in theory, it could retain infectivity on a flight or in a ship container. Yep. There's much less news about new SARS-CoV-2 variants relating to changes in clinical course. Is it because the clinical course will likely never be changed, or because nobody's looking? Well, all the studies on clinical course of variants are flawed. You can't make observational studies in people and say this variant is more or less pathogenic than another. Okay. There are too many confounding variables that you can't account for. And so people say, oh, Omicron is mild. Really? Really? It's killing a lot of people. It's mild? Where do you get that from? It depends what population you're in. So I think people are finally realizing that it's not useful. Delta is more virulent. No, in certain populations, maybe, but in others, it wasn't. And this is useless because it scares people. So people are finally realizing that, well, first of all, the variants are coming up so often, they don't want to do the studies anymore, but they're not useful. A big fraction of the population is vaccinated, and that takes care of a lot of pathogenicity, right? Bonnie took my mom to a doctor, masked, and doctor asked why in a rude way. Oh, that's absurd. That's not good at all. No, at a medical center, masking is absolutely acceptable, and a doc should be retrained on manners. My gosh. What people want to do is their own business. But in a medical center, it should be fine. Oh my gosh. Sorry. Red Hen is in southeast Pennsylvania. Elmug is from Pueblo, Colorado, where it's 87. Well, I think we should be using IPV, an activated polio vaccine, globally. It won't cause paralysis, and it will prevent it, and it won't transmit because it's inactivated. OPV should be eliminated. It should be in the U.S. Your OPV is delicensed. You can't use it for good reason, because it paralyzes kids. I don't care if it's 1 in 1.5 million. One paralyzed kid is too many. If it's your kid, right, then it's too many, right? So nobody in the world should be using OPV. Now, and Alan Dove says, well, if you don't use anything, then you're going to have paralysis. Yeah, but I'm not saying use anything, not to use anything. I'm saying use IPV. Oh, but it's a needle. It's hard to do. Figure it out. Figure it out. We figured out how to get COVID vaccines through needle to many people in the world. You could do it with IPV. Now, sorry. Marceline is from the Philippines. Hello, welcome to Virology. Maybe I'm getting bitten, but I don't react. I don't get welts ever. Mosquito bites. Never, never, never. Okay. So apparently Kathy Abscal is the writer here. Writes on nutrition in an accessible, non-preachy way. I should read it. What's okay? What do I read? I have to also read Vanity Nutrition's book. I'd like to do that. Yeah. Jane Crotty has a new video on mucosal immunity with Topol. Topol, the cardiologist, great person to ask questions about immunology. Forget about a nasal vaccine. Not going to be any better for reasons I'll tell you in the mini lecture. Tatiana is from Birmingham. Unlike in the UK, Birmingham. Yep, yep. It's interesting. So, Kathy is the author. Her book addresses the microbiome and sane, helpful nutrition. Cool. Very good. Welcome to the live stream. Yeah, memberships or subscribers will not reach the ones who don't want to hear scientific facts. That's it. You have to get them accidentally. Sean says, can we date bacteriophages by finding the CRISPR palindromes? I don't know about dating, but you can tell what hosts they infect by finding their arrays in bacteria. That's right. It's a good way to do that bioinformatically. Yeah. Will says, potential news. China CDC says COVID incidence is way down. Yeah, we're all having this big surge, as you know. And so what's going to happen? This surge didn't happen last summer. It's very interesting. Is it going to go down, and now in the late fall, early winter, is it going to go back up? Stay tuned. We'll let you know. I saw a guy masking on a sidewalk at least half a mile from many people. Well, you know, obviously that's not necessary, but on the other hand, maybe he was just zoned out and didn't remember, and he's just thinking, and that's okay. For COVID, what's considered frequent enough vaccination? Well, I'm going to do the mini lecture shortly, and you'll find out. Okay, and Martha's from Charleston, South Carolina. Hello, Amy Rosenfeld. IPV, there you go. Should be using IPV. But Vincent, cardiologists are people too. Yeah, but why is he interviewing an immunologist? To ask dumb questions? Why don't you have an immunologist or a virologist interview the immunologist? Because Topo wants the publicity. He wants the hits and the follows and the visible. He's seduced by, he's addicted to being a personality. That's all there is to it. I don't believe a word that he cares about you, because it's most things wrong. Okay. And so I don't want to hear what he asks Shane Crotty. They're going to be crappy questions. Stay in your lane. That's what it's all about. I have nothing against him as a person. Have you ever mentioned the bat virus HKU5 lag? Yes. Yeah, we've talked about it on Twiv. It's a bat coronavirus, and not concerning. Doesn't have pandemic potential, essentially. All right. Let's do a mini lecture. I've been thinking about doing this for a couple of days now. This is how I come up with my mini lectures. And because every time a new vaccine is announced, people get confused. What am I supposed to do with this? What does it do? Why do I need it? So let's do some basic immunology and give you some scenarios, okay? So let's start here. Antibodies, T cells, and SARS-CoV-2 vaccines. Not all. I'm just going to talk about vaccines in general, mostly mRNA vaccines, okay? But first, I want to do some basic immunology. I'm not an immunologist, but I know enough to talk about viruses and immunology. Just the overview, excuse me, the overview. Host defenses. We got three kinds, broadly. We have intrinsic, which are always present in the infected cell, like things like apoptosis, programmed cell death, autophagy, RNA silencing, antiviral proteins are always there. And if an infectious agent comes into the cell or organism, they're ready to work. We're not going to talk about those. And we have innate immune defenses, which are induced by infection. So you're infected with a virus, and within minutes, your body makes interferons, which are proteins that then turn on other genes that have antiviral properties. They're very important. Very important for, why do I have this guest here? I don't want that. Thank you. They're very important for getting through COVID. Interferons, the early interferon response is really crucial. If you make a bad response, you're likely to have severe COVID. And why would you make a bad interferon response? A lot of reasons. Everybody's, excuse me, everybody is genetically different. And in fact, about 20% of the population we know makes antibodies to their own interferons, which is bad, very bad. You're not supposed to make antibodies to your own proteins. And so that blunts your defense. And then finally, the adaptive system, which contains antibodies and T cells and B cells. And that is tailored to the pathogen, and it has memory. It's beautiful. It's beautiful. So we're going to talk mainly about antibodies and T cells today. Now, we talk a lot about antibodies, but do you know what an antibody is? Does anybody know what an antibody is? This is what an antibody looks like schematically. It's a big protein. It's actually got 4 chains, the protein. The 2 heavy chains, which are shown in red, and 2 light chains, which are in green. And they combine to make this Y-shaped molecule, which binds other proteins, and not just proteins, but other things, including proteins. And the way these are made is, let's look at this graph on the right here. Let's say you inject someone with a vaccine. We call that antigen A. Within 2 to 3 weeks, you start to make antibodies against that protein. So you see serum antibody titer on the y-axis is going up. That's because your body says, this is a foreign protein, and makes antibodies against it. Those peak, you can see here, it takes about 2 weeks, but maybe sooner, it depends. And then it peaks, and then they go back down again. These are serum antibodies. So they go up and down because you don't want to keep high levels of antibodies. Otherwise, your blood would be like jelly because you have antibodies against so many things. You can't keep them all at high levels, so they contract. And this, early on in the pandemic, was considered a defect of the vaccines, when in fact it is a property of the immune system. And if you just read, you would know that. But this is where cardiologists go wrong, right? Oh, the antibodies are going down. Oh, Mr. Cardiologist, did you know that's a natural function? Willful ignorance is not good. Okay, then you typically maintain antibodies at a low level. And if you see the antigen again, you make a really rapid response and a higher response. So within days, the antibodies go up and much higher, because now you have made memory B cells, which make the antibodies. And they recognize the antigen when you come back with it. And you have a quick memory recall. But look, it's not immediate. So you have low levels of antibody. You encounter this antigen again, antigen A. A couple of days it takes for it to ramp up. So you're going to get infected. You're never going to prevent infection. Maybe in this early phase here, if you first get vaccinated, within a month or so, you have high antibody levels, you're going to inhibit greatly infection. You're never going to completely inhibit it. It's just not possible. But just think, these antibodies, let's just focus on the upper tract. The antibodies are floating around in your mucosal layer. The virus is not going to be hit by everyone. So even with high antibody levels, you're going to be infected, but it will be less. You make less virus. You reduce the virus load. Okay, so that's the characteristic of antibodies. They are specific and they have memory. So before we go further, let's define the terms we're using here. So an antigen is a molecule that induces an immune response, like an antibody response. It can be a protein. It can be DNA. It can be RNA. It can be lipid. It could be polysaccharides, almost anything. Okay? That's an antigen. An epitope is part of the antigen that's bound by the antibody, or for T cells, T cells can recognize epitopes, the T cell receptor. So here's an antibody molecule on the right here. And you can see the protein with each sphere is an amino acid. And the epitope, that's the antigen, the protein, the whole blue thing. And the epitope are the few amino acids that are actually bound by the antibody. Epitope. And then we have a monoclonal antibody, which is against one epitope. So this is a monoclonal here. It's binding one epitope. Now, all antibodies are monoclonal, and what's in your blood is a mixture of lots and lots of monoclonals against individual proteins and different proteins, of course. So serum contains a mixture of monoclonal antibodies. We call it polyclonal sera because it's a mixture of monoclonals, okay? Now, in terms of virus infections, some antibodies do what we call neutralize. They block virus infection. That's an essential defense against many virus infections, right? If you have antibodies in your nasal mucosa, nasopharyngeal mucosa, they will reduce viral loads. They won't block it. They will reduce viral loads. I mean, this idea that just because you have a nasal vaccine, it's great, but the antibodies are going to contract, and it's going to take a few days for you to induce them. So it's no win whatsoever. No win whatsoever. Look at FluMist, the flu intranasal vaccine, no better than any other flu vaccine. So stop touting COVID nasal vaccines. Now look, I'm not saying you shouldn't try, because that's the mother of invention, right? You have to try, but don't say this is the solution, right? Let's say we don't know and we're going to try it. Anyway, so the antibodies are in your blood, they're in your tissues, organs, and so forth. They're also at your mucosal surfaces, and there are different kinds of antibodies, IgG, IgA. They can be at mucosal surfaces as well, okay? And the key is this last point here, not all antiviral antibodies neutralize infection. So when you make antibodies to a virus, some of them neutralize, some of them block infectivity, but others don't. Yet they can still be important, as you'll see. All right. All right. So this is a neutralizing antibody. This is an assay that you would do to say, I have neutralizing antibodies. It's a plaque assay. So you have a virus, and you have made a plaque assay. You can see on the bottom here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 plaques. Yeah, that's what the number means, Vincent. And plaque assay, as you know, is a way of enumerating infectivity of viruses in cells and culture. And on the top, we have mixed the virus with antibody and then did the plaque assay. So if the antibody blocks infection, you won't see a plaque, right? So here's a 1 to 10 dilution of the antibody, the serum. No plaques. All the virus has been bound by antibody. 1 to 100, still no plaques. Now at 1 to 1,000, we're getting a little bit of plaquing, 2 plaques. And then at 1 to 10,000, we have no neutralizing antibody. So that is a neutralizing antibody assay. If you have a new SARS-CoV-2 variant, you do this. You don't actually do this because people don't do SARS-CoV-2 infections normally, because you need a BSL-3, and not everybody has that. But you would do an infectivity neutralization assay to see if the new variant of SARS-CoV-2 can be neutralized by antibodies against elicited by a vaccine that's not the same as the variant, for example. Okay? So that's neutralizing antibodies and how we measure them. How do they work? Well, on the left, the antibodies can bind to the virus. You can see them binding to this virus particle here. They can block attachment. That's a major mode of function of neutralizing antibodies. But they can also block uptake of the virus into cells, the process called endocytosis. They can block genome release, Mike Strauss, genome release of the RNA from the particle. Mike Strauss doesn't like uncoding, so I'm using genome release for Mike Strauss, who is in Montreal, by the way, at McGill. Then they can aggregate the particles. So there are a number of ways that viruses can antibodies can neutralize viruses. And so on the right, this is where they are. So this is your nasopharynx here, upper tract and lower tract, e.g. lungs. And it's lined with mucosal cells, as you can see in this blow up here. And there will be antibodies present, well, in the blood circulation below the cells. Of course, here's a blood vessel. But the antibodies will make their way into the mucus layer on top of your epithelial cells, like I've shown here. They will encounter viruses there. And if the titer is high, the antibody titer is high, they can neutralize quite a few viruses and lower the frequency of cells that are infected. So they'll lower the viral load, they'll alter the kinetics of infection. But if there are no antibodies present, or if it takes 2 days for them to come on because of memory, and it's been over a few months since you had your vaccine or your infection, then you're going to get infected, right? That's the way it works. But you're going to eventually make antibodies and you're going to make T cells, and that is going to prevent severe disease. We'll talk more about that later. So that's how these work. What do they bind to? Epitopes, right? Antibodies bind to epitopes. We call these neutralization antigenic sites on the virus particles. And here's poliovirus. All these white dots are places where antibodies bind. Here's influenza HA, hemagglutinin, the glycoprotein of the virus. These colored regions are places where antibodies bind and neutralize infectivity. But again, remember, not all antibodies neutralize. Many of them will bind, but they don't neutralize infectivity. So you may say, well, why bother to make them? Well, you make antibodies against all parts of a protein. You don't have a choice. But actually, these non-neutralizers are useful, as I'll show you in a bit. Of course, we have lots of monoclonals against SARS-CoV-2 spike. That's the spike protein right there. And all these different colored regions are different parts of spike where we've made or isolated monoclonal antibodies. Right? The spike is the protein in the membrane of SARS-CoV-2, and it attaches to the cell receptor ACE2 via the receptor binding motif and the receptor binding domain. And this is where most of the monoclonals that neutralize infectivity bind. They block attachment by doing that. Here's a model of ACE2 bound to the receptor binding domain of Omicron. And you can see this interface between the two. Most of the monoclonals that neutralize infectivity bind amino acids at that interface. So this has been extensively studied, as you might guess, because we'd like to know the nature of the immune response. And for a while, we used monoclonals therapeutically. But then variants arose, and these were not bound by the antibodies. Now, what about non-neutralizing antibodies? We know that they can protect you against disease. So the idea that we measure just neutralizing antibodies and say, aha, your neutralizing titers are low, we must boost you, is inherently wrong. I mean, if you have neutralizing antibodies, they will give you some mode of protection, for sure. But if you don't have them, maybe you have binding but non-neutralizing antibodies or T cells, likely that you do, that can help. So here's an example of disease protection by non-neutralizing antibodies. Here's an antibody that is able to bind this virus particle, but it doesn't neutralize infectivity. It doesn't block infection by that virus. But now the antibody bound to the virus can bind to, in this case, it's a macrophage. And macrophages have receptors on their surface to which antibodies can bind. And you can see these antibody virus complexes are being bound, taken up into the cell and being degraded. So those antibodies are helping the cell to destroy the virus particle. So you don't need to be a neutralizing antibody. But here's the dark secret. people only study neutralizing antibodies for the most part because they're really easy to measure. It's hard to measure non-neutralizing antibodies. It's also hard to measure T cells. So people take the easy way out and they do neutralization assays. Now, I understand I'm not being fair, but it's the truth. The other assays are hard, so people don't do it. So that's how, one way. There are many ways that non-neutralizing antibodies can be helpful in protecting against disease. Now, viruses mutate, right? Of course they do, all the time. It's not a headline. And some of those mutations can lead to changes in those epitopes to which antibodies bind, and they will break the ability of the virus to bind. This is called evasion of antibody. And it happens all the time with influenza virus. Here's the HA. Single amino acid changes in these epitopes, the different colored regions where antibodies bind. Sometimes one amino acid is enough to break antibody binding, and that's enough to change the flu vaccine from year to year. We go, up, the vaccine-induced antibodies are not going to recognize this flu variant. Let's change it. That happens once a year for flu. We change the vaccine once a year, and it seems to be adequate. On the left, rhinoviruses do it differently. They exist as over 100 antigenic variants or serotypes concurrently circulating. And so you would get infected with rhinovirus 34. You get a mild cold, and then in 6 months, you get infected with rhinovirus 89. Okay? And they're antigenically different, so your immunity will not protect you against disease. And of course, SARS-CoV-2 spike underwent massive and is still undergoing massive antigenic variation. These are some of the sites on the spike that have changed, single amino acid changes that lead to evasion of antibody. Okay, so that's antibody. And then I want to talk about cell-mediated immunity, and then we're going to put it all together in the context of COVID vaccines. We have antibodies, but we also make cells that can counter virus infections. And this is called cell-mediated immunity, and a big part of it is the T cell. T cells, CD8-positive cytotoxic T cells, which recognize a virus-infected cell and kill it. So these CD8-positive T cells are generated during infection, just like antibodies are generated by B cells. There's a separate mechanism for T cells. They recognize a specific epitope from the pathogen, and they recognize that epitope on the surface of the virus-infected cell, and they end up killing the cell. So these T cells are really important for preventing disease. And one thing I want to say right now, SARS-CoV-2 variants may change, and they can evade antibodies, so the antibody won't bind. But none of the variants evade T cells. The T cells work from the first to the current variant. There's no change. And in fact, those are what prevent severe disease, everything else being equal, but everything else is not often equal, and that's the problem, and we'll see that in a moment. So remember that. We may have antigenic variation that allows viruses to avoid antibody. But for SARS-CoV-2, there is no variation that avoids T cell mediated immunity. And again, people don't measure T cell immunity very much, so it's not paid much attention to. So, the next variant comes out and we say, oh, yeah, the old antibodies don't neutralize it. We have to make a new vaccine. Well, the T cells are still going to kill virus-infected cells. So how do you know we need a new vaccine? And we don't. We do not know. We do not do the science to show that we actually need a new vaccine. And what we're doing is saying, we're not sure, but let's just induce a bunch of antibodies, and that's going to reduce virus infection. So that's the logic that we're going by. It's not good science. Now, these adaptive responses, antibodies and T cells, also give you immune memory, as I showed you in that first slide. You should know this painting. and the name of it and why I'm using it for this slide, right? There will be a quiz. If you notice that I've added virus particles to the clock faces. The memory is important because if you're infected later with the same virus or a similar virus, you'll have a rapid and specific response. I showed you the curve. It goes up faster and higher, the antibodies and T cells. And that's the basis for vaccination. We give you a vaccine that doesn't make you sick, and then you get memory. You make B cells and memory T cells, and then when you encounter the pathogen, you make a response. Delayed, yes. So you will be infected, you will get mild disease, but all things being correct, you will not get severe disease. Okay, so this is memory. First infection, you make an initial response. This can be antibodies, it can be T cells. They peak. So you're in a period here where you have a lot of these antibodies and T cells, say in your nasal mucosa. You're likely to have very mild disease after infection. These levels decline, and then at some later date, you get infected. Boom, you have a rapid response of antibodies and T cells. Not immediate, it's rapid, it's a couple of days. So you're still going to be infected. But the combination of antibodies and T cells will make disease mild. That's memory. And it's amazing for some viruses how long memory lasts. So this is a measure of the persistence of antibody. So they took people that had various diseases, as you can see on the left here, and they took blood and they looked for neutralizing antibodies against the different viruses that cause these diseases. And so these are grouped into systemic infections and mucosal infections. Look at this. Some of these are amazing. Yellow fever. Some people have antibodies for 75 years. And if you have antibodies, that means you have memory B cells. So they're making a little bit of antibody. It's a low level, but it's there. And if you got infected, you'd have a memory response and be protected. Measles, 65 years. Polio, 40. Smallpox, 40. Vaccinia, 75. Oh my gosh, these are really long. But then look at the mucosal infections. These are done, coronavirus is the common cold coronas. 12 months. After that, you don't detect antibodies. Influenza, 30 months. RSV, 3 months. Rotavirus, 12 months. This is pretty crappy, folks. The memory is not lasting very long, at least the B cell memory. For some of these, the T cells last longer. I know that SARS-1, some of the survivors had T cells 15 years, 17 years after being infected. Anyway, it's tempting to say that the systemic infections give you a better memory because the virus is circulating in your blood and it has access to many more lymph nodes. So it can establish big populations of memory B cells. Whereas the mucosal infections, the virus's reproduction is limited to the mucosa. Not so many lymph nodes there, maybe restricted memory cells. I mean, that could be a totally naive idea, but it's just one. You think of something if you don't like it. Okay. Now, let's end by putting this in the context of COVID and COVID vaccines. They got different scenarios here and outcomes. So here is your first infection with SARS-CoV-2. You're not immunized. You're not infected. It's your first one. So what's going to happen? You're going to have a delayed antibody and T cell response. It's going to take 10 days to 2 weeks to get peak antibodies and T cells. Right? Eventually, you're going to have antibodies and T cells in your respiratory mucosa. Those will, in best scenario, restrict infection. You'll have mild disease and you'll recover. Now, the problem is, if you're over 65, you're more likely to have severe disease because for many factors, and a big one is interferon. Interferons are incredibly important for early control of virus infection. If you make a really good interferon response, you're likely to have mild disease. If you make a crappy interferon response, the virus replication is going to shoot up. And then your adaptive response, the antibodies and T cells, are going to overreact. And then you're going to get your inflammatory disease. You're going to have low oxygen and you're going to end up in the ICU. That's 65 and up. Okay, so that's your first infection. All right, so now you're vaccinated, and the circulating virus is a good match for the vaccine, which means that the antibodies the vaccine generates can block infection by the circulating strain. Right. So if you've been recently vaccinated, I don't know, within the past months, you're going to have high antibody and T cells, right? And if you get infected, that's going to limit viral loads and progression. So you're going to have mild disease. But if you were vaccinated over a month ago or infected, doesn't matter. You're going to you're going to depend on a memory response, right? A memory response. And that's going to take some time, a couple of days, in which case the virus is going to reproduce. If you're young, you'll most likely be healthy. You'll recover because you can make good antibodies and T cells. But again, if you're over 65, not only do you have some issues with interferons, but since you first encountered the virus at an older age, you don't make good memory T cells. So you may make some antibodies, but it's the T cells that are going to prevent severe disease. And older people just cannot make good memory T cells. That's why the burden of severe disease is among over 65. The defect in interferon and the inability to make memory cells. Okay, so that's a good match. What if you have a variant that's not a match for the vaccine? So now any neutralizing antibodies you have, whether they're from a recent immunization or from a memory recall, they're not going to neutralize infection. And so your virus is going to infect throughout your upper tract. It's going to reproduce extensively, but you're going to have a memory T cell response, and that T cell will recognize the variant. It will clear variant virus infected cells, and it will prevent severe disease unless you're over 65, in which case you can't make good memory T cells. That's the rub of being over 65. So if you're under 65, and these dates are not absolute. It's not like at 65, boom, you get bad T cells. It's a gradient. Everybody's different. You're going to get infected. Below 65, you're going to get infected. You're going to make a memory T cell response, and that's going to prevent severe disease. Over 65, you won't make those memory T cells. You won't recall them because there aren't any. There are fewer, and the replication will go unchecked, and you'll have severe disease. So that's what happens with a variant. In theory, one day everyone will have been infected at a young age, and we won't have this T cell memory issue, and people won't die as much as they are dying now. And most of the deaths that are occurring in the U.S. are over 65, and most of them have comorbidities, which make the immune defense even harder. All right, and last, so what a matched vaccine does in theory. So now the CDC is saying get the latest circulating variant. Why would you do that? Because I'm just telling you the T cells are going to be there no matter what. If you had the original vaccine, those T cells will still protect you. Yeah, if you're under 65 and you made those memory T cells. If you're over 65, you likely didn't make them. So what are you going to do? Well, the idea is you take a vaccine every 6 months and you get higher levels of antibodies and some T cells, but not memory T cells, in your nasal mucosa. And if you get infected in the next month, it'll prevent mild disease. But, a month or so off, you're going to be depending on memory responses. So if you get today the latest vaccine variant, and in a month, you're still going to get an infection. And if you're over 65, it's likely to be severe because you don't have good memory T cells. If you're under 65, you're okay. Now, if I were 50, I would have gotten the 3, this is in hindsight, I would have gotten the 3 initial COVID vaccines, and that's the end of it. And I bet that would have prevented severe disease, assuming no other comorbidities and so forth. All right, so, but you don't want a COVID vaccine every 6 months. That's really crazy. Gosh, you don't do that with flu, although I guess over a certain age, you can get it twice a year. Be prepared to take an antiviral, right? If you test positive, take a Paxlovid if you can, and that will prevent severe disease. Contrary to what many physicians and healthcare workers say, it is highly effective at preventing severe disease. All right, so then other considerations here. Long COVID. Many people are afraid of long COVID. You can get it very young. You can get it from an asymptomatic infection. You can get it very old. So, everything I've said, if you're worried about long COVID, it doesn't help, right? And the other thing is that young people can get severe disease because we assume under 65 you're absolutely healthy, and not everybody under 65 is absolutely healthy. They have other sorts of comorbidities, and you can make generalizations, but it's never going to apply to everyone. So what does the CDC say? Because I'm not making a vaccine recommendation. I'm just telling you the biology behind it. What does the CDC say? Pretty complicated. So your first vaccination, if you're not immunocompromised, you're going to get 2 doses if you're young, 1 dose if you're older. That would be your first vaccination. If you've had previous doses, you're going to get 1 or 2 or 1 depending on their age. Look at this, an additional dose of the 2024-25 vaccine. So that's the latest update, just announced last week. For people 65 and older who are not moderately or severely immunocompromised is not currently recommended. Look at that. So if you've already had this dose, they say don't get more. I don't know how you could already have it, but. Okay, and then if you're immunocompromised, you need more doses to get a good immune response. All right, so that is what the CDC recommends. But I've told you the theory behind why we update. It's basically to make lots of antibodies for a short period of time and give you mild disease. But you cannot get around the fact that over 65, you have trouble making memory T cells, and they are the key to protecting you against severe disease. And that is not something that you will ever hear from Eric Topol. Okay, thank you. Please ask questions. I wonder if we have any. Let's go down a bit. Vaccination is cheaper than Paxlovid. Yeah, but you can't get vaccination every month, right? If you got COVID every month. Well, don't let cost dictate your health care, okay? Please. All right, so there were some questions here. Let's see if I can find them. Polio, Eastern encephalitis, chicken broth. Hampstead Heath, Milwaukee, staphylococci. Mendi. I forgot Mendi. Mahalo Nui Loa. From Hilo, Hawaii. Welcome. Welcome, welcome. I don't know how many people are actually worried about COVID or worried about long COVID or worried about vaccination. I don't know how many people get it frequently. Maybe just older people. Bobby said, I used to mask outside or in my car by myself because I had a habit of touching my face or nose and I didn't want to transmit. Yeah, that's a good point. That will prevent you from touching your nose. Yeah. Yep. What am I drinking? It's water. And I should have made a cup of tea, but I forgot. And, water is just not interesting. But when I talk a lot, I get a scratchy throat. Next time I should have tea. I agree. Would be much better. Thank you for your support of science communication, G. Ferraro. Maybe the person wearing a mask on the sidewalk was wearing it because they have allergies to pollen. Well, listen, I saw a wonderful post on Instagram today, and I'm going to relate it to you if I can find it. Yes, I can find it. Yeah, here we go. Everyone you meet is fighting a battle you know nothing about. Be kind. Okay? Before you start yelling at someone or complaining or getting annoyed, everyone has some kind of a battle you know nothing about. So be nice, always. Default behavior, just be nice. Don't be an ass. I try and do that every day of my life. Not always successful. Yeah, short-term antibodies, we all got infected. You got infected the same day, the next day, the next day. You get infected all the time. It's just the extent of viral load is different. Bonnie says, could high cholesterol induce an immune response? That's a good question of whether having high cholesterol or normal cholesterol is any different. I'm not familiar with antibodies to cholesterol. It's a good question. What is the mechanism from an antibody encountering a virus and more antibodies being made? So you have millions of B cells in your body, each of them with a single antibody on the surface for one epitope. And when you get infected with a virus, that B cell with that antibody finds the virus and binds to it, and that causes the cell to proliferate and make more, and then those cells eventually make antibodies. So that's just a rough way, a quick way of saying how it works. But it all has to do with that antibody on the surface of the B cell, which, you have millions of these B cells circulating in you, and each recognizes a slightly different epitope. But there are B cells in you that will recognize even synthetic antigens, things that don't exist in nature that humans make. So they're so diverse, and the mechanism for that is amazing, but we don't have time to talk about it tonight. But that's the mechanism. And then some of those become memory B cells, and they reside in your bone marrow. And then when you re-encounter the pathogen, they bind the pathogen and start to divide and make antibodies. Okay, Will, this is a good point. Nasal vaccines, natural infection comes through the nose, doesn't seem to give better protection than injected. Other things being equal, why should nasal vaccines be any better? I've been saying this for years. What's the reason? Yes, you have natural infection in the nasopharynx. You saw the immunity is not very durable. So, yeah, I think it's the idea may be that you could somehow design a vaccine that makes high levels of antibodies all the time, but that doesn't seem like a good idea. The immune system is designed to work as it is, to make antibodies, and then they go down. So don't mess with it. Then you're asking for trouble. Here we go. Veronika, Privyet. 10 degrees C in Moscow. Welcome. We've been doing a little Russian in your absence. Thank you, Gail. Glad you liked it. I hope you enjoyed that. Yes, the painting was Persistence of Memory by Salvador Dali. I love it with the clocks melted, and I just put viruses on them. I've heard that boosters reduce the risk of long COVID. Yes, It does. I mean, it's not 100%. It's a low percentage. We've talked about it on the clinical update. But so vaccination and boosters is just one way to reduce long COVID risk, and using an antiviral is another way. And, you add them all together, you get close to a good number. Persistence of antibody is not the whole story. I didn't say it was the whole story. First of all, the persistence of antibody reflects the presence of memory B cells, right? And the point is there's a delay between when they respond, so you're going to get infected. And then if there's a bad match between them and the virus will continue to reproduce, and then you're dependent on T cells. And how much antibody? Nobody knows. Nobody's done that experiment. With smallpox immune memory just 40 years, would anyone be immune to Mpox today? 79, right? No, probably not. Last people immunized. Oh, I'd be immunized in the 60s. I'm not going to be immune anymore. No. Les wants to see a mini lecture on FC-mediated effective responses. Yeah, that would be cool. Let me write it down. Mini lecture. Is this worth breaking out on its own or not? Tell me. Be honest. As a standalone, like I did the MERS thing. Is it useful? Mini lecture on FC in disease. Yeah, this is cool. Thank you. Thank you very much. Can't they invent T-cell memories? Well, this is a challenge, right? Could we design a vaccine that would stimulate T-cell memory in old folks? Or are we beyond help? I don't think we should give up. People should try it, right? It's a good idea. Over 65 T cells, poor memory. What about B? Cell memory B or better? Yep. Now, why is that, right? I don't know. But in general, the older folks have better memory B cells than T cells. How would we properly define antiviral antibody? Oh, an antibody that reacts with any epitope on any viral epitope. Could be on the virus particle, or it could be a protein that you only see in infected cells that's not in the virus particle. Antiviral protein to which antibody is directed. Yeah, because neutralization is a very specific subset of those antibodies. Yeah. Patricia, how does Paxlovid impact immune response? I remember doing a study with Daniel, reviewing it, and they found that the immune responses to spike were unchanged in patients treated or untreated with Paxlovid. So, when you get those first symptoms that make you test and then you start Paxlovid, you've already got a good amount of virus replication. So that's sufficient to give you your immune response. Why don't we have a similar T-cell response to flu? We do. You get vaccinated, you don't end up in the hospital with flu vaccine because of the T cells. They're very important for recovery from T cells. Yeah. Of the patients who died currently of COVID, has anyone broken them down? by attendance, comorbidities, vaccination, and access to antivirals. You know, I'm sure some of this has been done because I just remember with Daniel reviewing um, cases, and I think on a recent clinical update, like what kind of things the people went to. So, um, yeah, um, some of the studies also, I don't know about access, but using antivirals, right? That's a different question. So, for sure, vaccination status, sure, comorbidities. I mean, these have all been done in separate studies, not all together. But yeah, many of them have been looked at. How do you account for people who are who don't get infected? Well, there's always a problem that you don't know, right? Maybe you are infected, you just haven't looked. um, I mean, I think it's all about immune responses, right? And there's a wide range. So people have antibodies to their own interferons, and then there are people who don't, and they make good amounts. There's some people who respond really well to infection with interferon, and other people who don't. And so people who may not get or seem to get infected probably have a really good interferon response initially. And yeah, so there are people like in families of multiple people, say 6 people, one person comes and gets infected, nobody else gets infected. Maybe the rest of the family was immunologically good, but hard to study. So we only speculate, right? Is there any pathway for upping numbers of IgG? Is there an IgG shot? Well, yeah, right. Giving people plasma, convalescent plasma. You're basically giving them antibodies that can supplant whatever they're not making. Um, but, and the many monoclonals were given to people early in COVID to try and prevent infection. But um, it's not a, it's not something that you want to do on a scale, right? Sorry, I get the vaccine with flu shot. Don't be sorry. It's fine. I'm just trying to explain the science behind what's going on because I thought people don't understand why we have to do this. So I'm trying to explain. But what you do is fine. Don't be apologetic. Okay, Eric says, would you need vaccination every month? No, no. Vaccination every month is absurd. I mean, twice a year, I think, is a lot. I just don't think we need to chase every variant. Right? In June, they made a recommendation. Now they've changed it for this fall's vaccine update. There's no evidence that it's beneficial, clinically beneficial. It's the theory that if you have antibodies, they're going to reduce infection, right? They're going to reduce the severity of disease. That's the theory. Yeah, that makes sense because neutralizing antibodies, if present, will neutralize virus, right? I'd like to see the data saying it's clinically useful. Why? I don't think you should do things because you have a hunch or it doesn't hurt or whatever. I think you should have evidence-based reasons to do it. And we don't. We don't. Oh, I'm sorry, Lori got COVID on her last flight. I'm sure you'll be good. Take care. I can't recall. Can plant viruses infect mammals? No, not usually. No, not usually. Welcome, Sam. Welcome. I had an older patient test positive for pertussis last night. Wasn't sure if I needed to be any more careful than any other virus. Well, it's a bacterial infection, right? Yeah, I mean, these are respiratory pathogens. So you need to have that person mask and not to infect anyone else. Least though in Australia, masks are back at the supermarket, mainly influenza. It's good. Good to see it. Should over or under 65 with EKG changes after the first or second dose be encouraged to get the booster? You mean associated with vaccine administration? I don't know. I wouldn't want to say. I think Daniel should answer that. Can you send it to Daniel at microbe.tv? I'll have him answer it tomorrow. Dagfin, the amount of antibodies reflects the amount of activated B cells and plasma cells. Yes. If these are inadequate, immunity is compromised. Very simply put, the whole story is a lot more complex. Of course, it's more complex, Dagfin. This is the purpose of science communication, to try and simplify things so that people can understand it. So, but this is not the point here. If you have memory, this doesn't matter because the antibodies are going to eventually appear and assist, right? Standalone mini lecture. Okay. What makes when one makes a strong interferon response, does that person feel like they've been run over by a truck? Maybe. It's in some yes, in others no. It really varies from person to person. But yes, when you make a good interferon response, you get inflammation, and that's the run over by a truck feeling. Yeah. So neutralizing means preventing attachment mostly, but there are other mechanisms. For SARS-CoV-2, it's mostly preventing attachment, but for other viruses, there are other mechanisms. So I would say neutralizing means blocking infection. Wasim from Kashmir. It's autumn. Session on lymphocyte trafficking. I don't want to do that because it's not my field. Get to Brianne Barker to do it. Maybe we get her here and she can talk about it. Hello, Siddhartha. Welcome from New York City. Thank you for your support of science communication. Really appreciate it. Yeah, that's the thing. If you never got COVID, so you never got sick, but you tested and never tested positive. But maybe another time you didn't test because you didn't feel anything or whatever, and you were positive. So it's hard to know for sure, right? You would have to look for antibodies to the N protein, the nucleocapsid protein, to see, because that's not present in the vaccine. So that would tell you if you've ever been infected. Yeah. Paxlovid is not recommended in people whose renal function is severely impaired. Well, it depends, right? So that's statins also are contraindicated renal function. So there are certain issues, but not just age. You can be 80 plus. Tony Fauci took Paxlovid, right? He's 80 plus. So it's specific things, not just age, right? You're bit by bit, you're educating me. The thing is, Will, I know it's hard. The words are hard. The concepts are hard. Most people are willing to say, screw this. I can't take this. I'm not listening. I'm going to go listen to music, and I don't blame you. It's fun. But if you're patient and you just keep coming back, eventually you're going to start getting it, and you're going to feel really good. Like, oh, gosh, I get this now, and I didn't get it 6 months ago. So that, for me, it's the best thing I can do for you. Does fever increase the antiviral effect of interferon? The idea is that high temperatures of fever in themselves are antiviral. So, yeah, combined with interferon, it would be additive, I suppose. But I have issues with some of these conclusions. Yeah. I still don't understand why we get a yearly flu shot if it's all about T cells. Because in older people, they make crappy T cell memory, right? 65 and up. And if you are younger, you probably don't need it, but it's just a blanket recommendation, everybody. So that's my take on it. Lisa, so far I've not had COVID that I'm aware of, and I have been majorly exposed. Check antibodies to N. You know, if you got antibodies to N, you've been infected. Now, COVID is a disease, so that you say you haven't been sick. Have you tested? Have you tested a lot? Anyway, it doesn't matter. The definitive thing is to get antibodies to N. What are the death rates in countries with high and lower rates? I don't know. It's a good question, but you could find those data out there. Let's take a look quickly. Sometimes a quick Google tells you a lot. Of course, I get everything in the US first. Yeah, you know, in the US, oh my gosh. So in the US, deaths per 100,000 people. Okay. I should just bring this up. Let me do that. So here it is, COVID death rates by vaccination status. Now, obviously, early on, look, deaths per 100,000, 33 in the unvaccinated and 3 in the vaccinated. Who the hell says vaccines don't work? What's your brain? Jesus. Talk about stuff that matters. Anyway, here today, deaths per 100,000 people, unvaccinated 0.84, vaccinated with bivalent 0.17. So again, a decrease. So this is what I just found quickly, right? But you asked a different question. I realize that. But I would guess it's tracking in a similar way, that where you have vaccination, you're going to have lower death rates. It's substantial, right? Oh, okay. Sorry, I misinterpreted. The amount of antibodies is not something to worry about if you are immunocompetent and have immune memory. I may not. Oh, yeah. Of course. I agree. I mean, obviously the T cells are going to really control the severe disease outcome, and they are recognizing all the variants, right? So we don't have to worry about this changing. And in fact, in 50 years, everybody will have been infected at a young age. Everybody will have good T cell memory. There'll be very little severe COVID. This will be a common cold. Right now, we're in a transition period where we have a lot of people getting infected over 65, and they have to go. They don't have to go. You get what I'm saying? Yes. Good. Thanks for clarifying. Lymphocyte trafficking sounds illicit. Very good. I like that. Now let's see. T and T cell is thymus, and aging thymus is replaced by fat and disintegrating structures. There is 95% new produced T cells. So yeah, your thymus involutes as you age, and that may be in part responsible for lower memory T cells. Yeah. Can we use rhinoviruses to increase interferon releasing? Well, they will make you sick, so I'm not sure you can do that, right? I'd love to see Vincent reacting to TWIV 2020s. Oh my gosh, that would be funny. I think the very first episode where we talked about COVID in January is very funny. I think Microbe TV needs a podcast about medicine and public health. Yeah, I do. I think it's a good idea. I'm thinking about it. Amy reacts would be salacious. Oh boy, that would be fun. Negative home tests indicate that you're not infected. They do not prove. Yeah, positive test. So if you have antibodies and it shows you that you have been infected. If you don't, it supports that you're not. It's hard to prove a negative. Yes, this is the problem with science. Yeah. Are there any home tests for antibodies to N? Let's see. I think so. So here's a test at home, elicity, but what is it looking for? It's an at-home test. I don't know what the antigen, what it's looking for antibodies against. There's another one that says they're looking for antibodies to SARS-CoV-2 to see if you previously had infection. You know, they're not breaking it down. So there are home tests, whether or not they're for N. They must be, though, if you want to know if you're previously infected. Yeah. Death rates in various countries. There are so many variables. Absolutely. I totally agree that that's very hard. It's much easier in a country like the U.S. to do that. Yeah. So that's hard. You'd have to age match vaccinated and unvaccinated. You'd have to match other aspects of the population. Very hard to do. Yeah. Is there any advantage to using Novavax? So, you know, the short durability of the mRNA vaccines may be a function of the way they're designed, the mRNA, and it could be that the protein-based vaccines have better durability, and that's a good question. The studies haven't been done that would address that, right? Whether protein-based would give you a longer memory recall. But many people I talk to suspect that that would be the case. So, you know, in the absence of clinical data, and I doubt we're going to get any, if you can get Novavax, there is no negative. It's as protective as mRNA that just may be long-lived, right? It's not going to be less. And if it's the same, no loss. If it's longer, it's good. Artemis says, I was told the immune system makes a wider variety of antibodies with Novavax. I don't know what you mean by that. Against more epitopes? Let's see. I don't know that that's correct. I'd have to look it up, and I'm not finding it. I'd be careful about that. There is evidence to suggest that Novavax might be less painful. It could be because you're getting a lot of, in the mRNA vaccines, there's a lot of RNA, right? Physical amount, and to achieve the immune response. And for protein, you need much less. So it's likely you're going to have less inflammation, yes, with Novavax. Yes, U.S. government is allowing people order free test kits this fall. That's correct. But you know, with Novavax, you still get side effects because you're getting inflammation. That's what makes a good immune response. So, you know, everybody is different. So it's really going to be hard to compare to answer your question. Is it going to be easier for me to take? It's been a great office hours. I hope you will keep doing mini lectures. I will. I will keep doing mini lectures. You know, Patricia, there's overall there should be fewer, but you're still going to have side effects, pain at the injection site, right? Swelling, because that's the immune response doing its thing. But if it It's overall less than mRNA. I wouldn't be surprised because there's a lot of mRNA in there, right? It causes a lot of inflammation. Let's see. Lipowski says Paxton had a strain of bacteria called Streptococcus anginosus that developed into an infection and spread to the brain. Is a virus involved in getting it into the brain? I'm not aware of it. I'm not aware of any case where a virus helps something else get into the brain. All right, folks. If anyone goes through the 2020 episodes, the Ralph Barrick one is excellent. Yeah, Ralph nailed a lot of things on early. Joe DeRisi has been on TWIV. You can search TWIV. I'll do it for you. TWIV DeRisi 196. Wow. TWIV 196, Joe DeRisi. Yeah. In theory, should expired COVID tests work on today's virus? Yes. You know, they qualify them as long as they have tested out. And the date is as long as they've tested. But in fact, they can last a lot longer, right? You know, you just make sure that the lines are all right, the test line and all that. Oh, my professor loves TWIV. We have to write on a podcast of our choosing. Well, I hope you enjoy it, right? I have to say, I love doing them. All right, folks, thanks again. Let me thank everyone, all our moderators. So we had Les, we had Vanity Nutrition, we had Frank, we had Tom, we had Steph, we had Andrew. Thank you all for moderating tonight. Really appreciate it. You guys have done a great job with the likes. The likes are pretty much near the people who are here. And thanks all of you for coming. And we'll see you next week. So the next episode will be here next week. That is, what's the date? Wednesday, the 11th of September, 9/11. I'm sorry that we have to do one. However, we will have a special guest. Daniel Griffin will join us next week, September 11th, 8 p.m. Eastern. And you can bring your questions about medical things, because I don't give you medical advice. Bring them for Daniel. All the questions that came up tonight, any others. Daniel Griffin, September 11th, office hours, 8 p.m. Eastern time. I want to thank everyone for coming, and I want to make sure that you stay safe. Stay safe until next week. Love you all. Thanks for coming. Good night.